the cytoskeleton is what helps give a cell its shape and helps it also move around. So we'll be discussing three main components within the next few videos. So the first one is the intermediate filaments. Now these filaments are, first of all, they're monomer. They're made up of this alpha helix. So it's one alpha helix. Usually they're all made up of similar amino acids, and they're usually about the same length. So you usually also end up with the same diameter of your filament. So like any protein, they have a C terminus and an N terminus. So this is the monomer. And then when you want to form an actual filament, first you got to get two dimers. So dimers, you would have two of them, both C terminuses on the same side and then N terminuses on the same side. And then they become coiled until you form your dimer. And then each of those dimers is going to pair up with another dimer to form a tetramer. So remember that we are using these intermediate filaments for structural support or mechanical strength. So we want to build them in a strategic way so that it would give us that strength. So say you're trying to build a wall or something. When you're laying your bricks, you don't lay them one on top of the other perfectly like this, because that wouldn't be the strongest way to do it. Rather, you would put one and then the other, and you would stagger them in a way that they're overlapping, kind of like this, to give it more strength. So we do the same thing here. So when you take two dimers, you don't place them directly on top of each other, but slightly staggered like this, so it makes a tetramer. And then when you get 8 of these tetramers, each staggered all together, you make a intermediate filament. So these filaments usually all have similar diameter size. And then let's look at something that a microfilament actually helps do. So if we're looking at cells within, say, epithelial cells in your intestines, it's important. So when you eat food, you need to be able to stretch your intestines, right? Now, intermediate filaments kind of stretch out and go into neighboring cells through these connections called desmosomes. So in this picture over here, you see that the filaments stretching out and holding the cells together. So now if it stretches, that's okay. They're not going to rip apart and separate the cells because you have this filament keeping them together, again, providing that structural strength. Okay, so there are many different types of intermediate filaments. We split them into 4 classes, 3 of which are found within the cytosol and 1 is in the nucleus. So the 3 cytosolic intermediate filaments are keratins, which is the most diverse protein that we have within our bodies. So it's mostly found in epithelial cells, but there's lots of different types of epithelium, whether it be in your tongues or in your skin or the airway tracts within your lungs or intestinal tracts. They're all different types of epithelium, and each different type has a different type of keratin. So these keratins are an example of intermediate filaments, and we already looked at how they're important so that they give it strength, especially in structures that have cells that undergo pressure or any kind of stretching or anything like that. So they would need the strength to be able to stay together. For example, your skin, so that you don't get a bruise each time you hit it against something or stretch it a bit. And then after that, there's the vimentin, and that is in connective tissue, muscle cells, and also some neurological cells. And there is the neurological filaments, or the neurofilaments, which are very important because neurons do not undergo mitosis. You can't have more neurons after a certain amount of time. You only have that amount of neurons for the rest of your life. So it's important to keep them intact. And the way we do that is by giving them strength, especially in this long axon, which is very thin and could be fragile if it didn't have these intermediate filaments giving it strength. And then after that, in the nucleus, we have the nuclear lamins, which is found right underneath the nuclear envelope. And those are important. They have an important role during mitosis because actually they're going to get broken down by mCDK, remember, which is going to trigger the breakdown of these nuclear lamins, which then breaks down the nuclear envelope so that you can undergo mitosis, and then after that, reform the nuclear lamins once again when you're making the new nuclear membrane.